Hello and welcome to another Microbiology Bio120 lecture. This lecture is going to be about the nitrogen cycle. This is the image of the nitrogen cycle from your book. It shows all the different processes that we will cover in this lecture. Remember that the nitrogen cycle, similar to the carbon cycle, consists of redox reactions in which different nitrogen compounds are oxidized and reduced. Microorganisms are doing this to derive energy and also to form cellular molecules like proteins. The nitrogen cycle is distinct from the carbon cycle in that every process that you see, with the exception of assimilation and the release of waste, is exclusively mediated by microbes. In the carbon cycle, multicellular land plants also complete photosynthesis, and lots of multicellular organisms, all multicellular organisms, respire. However, only microorganisms can complete nitrification and denitrification. Only microorganisms can complete anammox, and so on. Here's a table listing the major processes that make up the nitrogen cycle and some of the organisms that carry out these processes. For example, nitrification is actually a two-step process, and Nitrosomonas is capable of turning ammonium into nitrite, while Nitrobacter is capable of oxidizing nitrite into nitrate. So different organisms are responsible for carrying out different steps in the nitrogen cycle. Here's another image of the nitrogen cycle, and you can see on the right there that the oxidation state for the various nitrogen compounds is listed. With this cycle, it is fairly easy to see which processes are oxidative and which are reductive. You should see that nitrification is the stepwise oxidation of ammonium to nitrate, while denitrification is the reduction of nitrate to dinitrogen gas. Note that many of the intermediate steps here are left out for this diagram. Recall that the Redfield ratio tells us the molar ratio of carbon, nitrogen, and phosphorus in microbes, and based on this, we know that organisms need nitrogen. Nitrogen is found in amino acids. In a lot of ocean systems, usable forms of nitrogen can often be limiting, meaning that if there was more nitrate or ammonium available, then phytoplankton would grow more and the system would be more productive. So I said if there was more nitrate or ammonium available, then organisms would grow more. There's always plenty of dinitrogen gas, but that isn't usable by most organisms. Dinitrogen gas, N2, makes up 78% of our atmosphere, but not a lot of organisms can use it. The triple bond between the two nitrogen atoms takes a lot of energy to cleave, and most organisms simply can't afford it. However, if you live out in the middle of the ocean and the only thing preventing you from growing more is a lack of usable nitrogen, then breaking that triple bond might be a good choice. Taking dinitrogen gas and turning it into ammonia is called biological nitrogen fixation, and the organisms that carry this process out do so when there are no other forms of nitrogen available, such as nitrate or ammonium. Biological nitrogen fixation is carried out by diazotrophs, and they can be free-living, either aerobic, like many types of cyanobacteria, or anaerobic, such as some of the anoxygenic photosynthesizers that are listed here. And they can also be free-living associative, such as Azospirillum, or they can be symbiotic, such as Rhizobium. So like I said, some types of diazotrophs form a symbiotic relationship, and some do it with multicellular plants. So Rhizobium infects root nodules, where they live mutualistically with the plant they have infected. It is a mutualistic relationship, in which Rhizobium provides the plant with fixed forms of nitrogen, usable forms of nitrogen, that it can use to grow, and the plant provides Rhizobium with glucose, so that it can respire and have enough ATP in order to cleave that triple bond in dinitrogen gas. But not all diazotrophs are symbiotic. Some are free-living, and if you're a free-living diazotroph, you have to meet the ATP requirement of fixing that dinitrogen gas into ammonium all by yourself. This explains why a lot of free-living diazotrophs are photosynthesizers or autotrophs. They can generate their own sugar so that they can break it down when they need to in order to meet that ATP requirement. But earlier we sorted free-living diazotrophs into aerobic organisms and anaerobic organisms. So why did we do that? We sorted diazotrophs into aerobic and anaerobic because nitrogenase, the enzyme that fixes dinitrogen gas, is irreversibly damaged by oxygen. Nitrogenase is a complex made up of two proteins that depend on having an anoxic environment for their redox reactions to be carried out successfully. You have dinitrogenase reductase, made up of iron on the left there, that is reduced by ferredoxin with a large input of ATP. It then reduces the dinitrogenase, made up of iron and either molybdenum or vanadium, which is then used to reduce N2 to ammonia. There are a lot of redox reactions that go into the fixation of nitrogen, and nitrogenase carries them out. The overall reaction is still dinitrogen gas to ammonia, then the ammonia is quickly converted to biological nitrogen. Because oxygen damages nitrogenase, diazotrophs that are oxygenic photoautotrophs have several strategies to deal with this fact. The first is spatial separation. Some types of cyanobacteria will live colonially, and they'll have certain cells that carry out oxygenic photosynthesis, so they can meet their ATP requirement, and other cells will be responsible for nitrogen fixation. The cells responsible for nitrogen fixation are called heterocysts, and they have thick walls that prevent oxygen diffusion. Another strategy for diazotrophs that are also oxygenic photoautotrophs is to temporally separate their activities. Some cyanobacteria will only fix nitrogen at night, since oxygenic photosynthesis requires light to proceed. Another strategy is to respire a lot in order to use up all the oxygen that's in the cell. This strategy also helps with producing enough ATP in order to meet the energetic requirement for nitrogen fixation. It is also possible to produce a protein that reversibly binds to nitrogenase that protects it from sudden oxygen stress. This is a fairly common strategy for soil nitrogen fixers. And finally, one of the best strategies for preventing oxygen damage as a diazotroph is to simply live where there is no oxygen. This is obviously the approach that the anaerobic nitrogen fixers and who are anoxygenic photosynthesizers use. This table shows some estimated rates of biological nitrogen fixation for different groups of diazotrophs, and I would encourage you to look at it more closely to get a sense of how much usable nitrogen is introduced to different systems by diazotrophs. This table is based on the previous slide, and as you can see, Rhizobium, which is the mutualistic diazotroph, they're contributing the most nitrogen worldwide to ecosystems. This is a function of the fact that they are being supplied with ATP by plants. It is important to remind ourselves what nitrogen fixation accomplishes. Nitrogen fixation takes dinitrogen gas, which is not a nutrient, and it turns it into a nutrient, ammonia. Ammonia is quickly taken up into biomass. The organic nitrogen that is in the biomass of the organism can then be remineralized by heterotrophic microorganisms and turned into an inorganic nutrient, such as ammonium, which can then be taken up by another organism, and the nitrogen cycle has started. So now we've covered nitrogen fixation, nitrogen uptake into a cell, and remineralization back into inorganic nutrient form. Now let's talk about nitrification. Nitrification is a chemolitho-autotrophic process. Nitrifiers oxidize ammonia or nitrite in order to gain energy. The ammonia can come from N2 fixation, nitrogen fixation. It can come from remineralized organic nitrogen, or it can come from a process called dissimilatory nitrite reduction to ammonium that we'll talk about later in this lecture. Nitrification is a two-step process. The first step actually consists of two steps itself, ammonia oxidation and hydroxylamine oxidation. The second step is nitrite oxidation. Both steps require oxygen to proceed, thus nitrifiers are obligate aerobes. Step 1 is carried out by ammonia oxidizing bacteria and ammonia oxidizing archaea. Step 2 is carried out by nitrite oxidizing bacteria. Nitrite oxidizing bacteria are often found living in close proximity to ammonia oxidizing bacteria and archaea because they depend on the products that they produce. Step 1, or ammonia oxidation, is the rate-limiting step. A lot of factors limit it, including the low energetic yield, which causes these organisms to be outcompeted by heterotrophic and photosynthetic microbes that also utilize the ammonia, in addition to the fact that it depends on oxygen, which is often limited in environments. Additionally, light can actually inhibit it, and this process requires a relatively narrow range of pH to proceed. Because step 1 is limiting, the product, nitrite, is typically used by nitrite oxidizing bacteria as soon as it is available. Here is a view of what is going on in the cell during that first step, along with the enzymes carrying it out. Ammonia oxoreductase and hydroxylamine oxoreductase. You can see that electrons are pulled off of reduced nitrogen species and funneled through electron transport chains, eventually reducing oxygen to water. A proton motive force is created, and with that, ATP is generated. Here is a similar view of what is going on inside the cell for nitrite oxidation. Nitrite oxoreductase is responsible for pulling electrons off of nitrite. Oxygen is again reduced to water, and again the electrons are used to create a proton motive force and generate ATP. Nitrification is needed, or has to happen, in order to create nitrate. And then nitrate is needed so that denitrification can occur. Denitrification, which you can see in that box there, involves many steps that are carried out by different enzymes. Complete denitrification starts with nitrate, which is reduced to nitrite, which is then reduced to nitric oxide, which is then reduced to nitrous oxide, which is finally reduced to dinitrogen gas, effectively undoing the work of nitrogen fixers. You can see the enzymes responsible for carrying out each step above the arrows, and some of the genes that code for those enzymes below the arrows. Denitrification is distinct from dissimilatory nitrite reduction to ammonium because the products are different. Denitrification produces N2 gas, while DNRA, dissimilatory nitrite reduction to ammonium, produces ammonium. Here is a visual for the enzymes that are involved in aerobic respiration and in heterotrophic denitrification. In the top right-hand corner, you can see where nitrate reduction subs in for the second set of enzymes. All laid out on the bottom are the enzymes in order, so you can see how the electrons are shuttled around. So here's our simplified version of the redox ladder, showing strong oxidants on the top left-hand side of the line, and strong reductants on the bottom right. You can see that combining denitrification, or combining nitrate with organic matter, so using nitrate as the oxidant, provides almost as much free energy as oxygenic respiration. So heterotrophic denitrification provides the organism with almost as much free energy as aerobic respiration. However, denitrification can also be coupled with the oxidation of other compounds as well. For example, the oxidation of hydrogen sulfide. This is an example of autotrophic denitrification. Similar to how you see nitrite oxidizers living close to ammonia oxidizers, you will see denitrification close to nitrification because denitrifiers depend on the products of nitrifiers. They need that nitrate. Just keep in mind that nitrification requires oxygen to proceed, while denitrification requires a lack of oxygen to proceed. So the two processes will take place on opposite sides of an oxic-anoxic interface, displayed here in a stratified water body. Both nitrification and denitrification are not 100% efficient. During both processes, nitrous oxide and nitric oxide can escape. Both are gases, and so they tend to escape to the atmosphere. Nitrous oxide, also known as laughing gas, is a potent greenhouse gas. It is stable in the lower atmosphere, but in the upper atmosphere it reacts with sunlight to form nitric oxide, which destroys ozone. Nitric oxide is a free radical, and NOx pollution, which includes nitric oxide, reacts to form smog, acid rain, fine particulates, and ground-level ozone, which is pretty bad. So human activities that increase microbial rates of denitrification and nitrification have global consequences. So now we've talked about nitrogen fixation, nitrogen uptake, nitrogen remineralization, nitrification, and denitrification. So the next step in the cycle is dissimilatory nitrite reduction to ammonium, or DNRA. It is a competing process to denitrification in terms that both processes utilize nitrate or nitrite. So DNRA is the reduction of nitrite to ammonium, though it can start off with nitrate and it will follow the same path as that first step of denitrification just to get it down to nitrite. You can see that nitrite reductase is utilized in order to reduce nitrite to ammonium, but it's a different type than what is used during denitrification. It's a different enzyme. It is distinct, this process is distinct from denitrification because the end product is ammonium rather than dinitrogen gas. So the nitrogen remains as a nutrient. It's able to be used by other organisms via nitrification or to uptake. This process is also distinct from organisms that reduce nitrogen in order to assimilate it into their bodies. This process can be either autotrophic or heterotrophic. It just depends on what the organisms are oxidizing. So some organisms have been observed to use this pathway, DNRA, in order to oxidize reduced sulfur compounds in sediment. However, this process could also be used to oxidize organic matter. You should notice that DNRA is not as energetically favorable as denitrification. Denitrification is right under aerobic respiration there at the top of the redox ladder, while DNRA can be seen several spaces down. But the fact that the nitrogen stays in a bioavailable form, in nutrient form, following its reduction, is a definite plus for this type of metabolism. We've talked about nitrogen fixation, nitrogen uptake and remineralization. We've talked about nitrification, denitrification, and DNRA. So now let's talk about anammox. Anammox, also known as anaerobic ammonium oxidation, is similar to denitrification in that dinitrogen gas is the end product, and it also happens in anaerobic environments. However, unlike denitrification, nitrite is used to oxidize ammonium. Additionally, it is exclusively a chemolitho-autotrophic process, whereas denitrification can be a heterotrophic process or autotrophic process. Anammox is performed by bacteria in the Planctomyces phyla, and it was first discovered in a bioreactor in the 1990s when scientists couldn't figure out why there was less usable nitrogen in the system than what there should have been. An intermediate nitrogen compound that is formed during anammox is hydrazine, which is also known as rocket fuel. Hydrazine is a very strong reducing agent. Because of this, planctomyces have a special chamber inside their cells called the anammoxysome. Its function is similar to that of mitochondria in eukaryotes, but it is also where the hydrazine is kept so that it doesn't react with other things in the cell. Now we've talked about all the different parts of the nitrogen cycle that we were aiming to talk about at the start of this lecture. And now you can refamiliarize yourself with some of the common microbes that carry out the different processes. But remember, this is not an exhaustive list. There are many organisms that carry out all of these processes. So if we were to think about the nitrogen cycle in terms of usable types of nitrogen cycling around, our inputs for the nitrogen cycle are just nitrogen fixation, while our outputs are the processes that produce N2 gas, something that not all organisms can utilize. And so our outputs for the nitrogen cycle will be denitrification and anammox. though nitrogen burial and ammonium adsorption could also be considered outputs. Note that neither inputs nor outputs mentions biological assimilation or remineralization. Those processes take nitrogen out of the nitrogen cycle for a little while, but it isn't as permanent a removal as the other types of outputs. So now we have to ask ourselves, how have humans changed the nitrogen cycle, and have we changed the nitrogen cycle? Well, a big way that we've changed the nitrogen cycle is by increasing the amount of nitrogen that can be cycled via the Haber-Bosch process. Humans figured out a way to fix nitrogen artificially during World War II by heating nitrogen gas and hydrogen together. This process provided ammonia for the production of explosives during World War I. Today, it is mainly used to create fertilizers, which we have steadily been increasing our use of. Recall the leaky pipe model. When we load more usable forms of nitrogen into a system, we also increase all the processes in the nitrogen cycle. We increase the rates of nitrification, denitrification, and anammox. Recall that nitrification and denitrification are not 100% efficient. And so if we increase nitrification and denitrification, we will also increase the release of nitrous oxide and nitric oxide. In addition to increasing the rate of nitrification and denitrification in ecosystems when we apply fertilizers to croplands, not all the fertilizers will stay in the soil. Some will enter groundwater, and if it stays as nitrate, that can be especially bad. The nitrate can come from ammonia that's oxidized, or it can simply come from the nitrate in the fertilizer itself. But wherever it comes from, increasing the amount of nitrate in drinking water is incredibly dangerous to our health, especially the health of infants that can suffer from something called blue baby syndrome. We think that about 1/3 of California's wells that supply drinking water are contaminated with nitrate. Another way that fertilizers interact with microbial metabolisms in natural systems is via runoff into natural streams. Remember that nitrogen is often a limiting nutrient in many systems, and so when that fertilizer runs off, a phytoplankton bloom can result. Systems that have plenty of all types of the nutrients they need are eutrophic systems. Changing the nutrients can change everything. When you add lots of nutrients and you have enough light, you'll have a phytoplankton bloom. Phytoplankton die and sink. Once at the bottom of a pond, heterotrophic microbes will consume the phytoplankton. Heterotrophic microbes will use up all of the oxygen at the bottom of that pond or lake, and so multicellular organisms that may need oxygen, such as fish, will die. Heterotrophic microbes can switch to alternate terminal electron acceptors, such as nitrate or sulfate, and eventually things like hydrogen sulfide and methane will start to be produced. This process doesn't just happen in ponds or lakes. Fertilizer runoff can affect large swaths of the ocean. This is a satellite image showing a dead zone that exists in the Gulf of Mexico because fertilizers increase phytoplankton growth and then subsequently heterotrophic metabolisms at depth. That concludes this lecture.